So the question is whether the observation of different polymorphs with different molecular structures in fibrils formed by the same peptide, whether this is a violation of the Anfinsen principle that the sequence determines the molecular structure uniquely, and it's it's not a violation. It doesn't violate any laws of physics or thermodynamics or something. It's just the simple fact that there are many different ways that a peptide can form a fibrillar structure that is sufficiently stable that it persists. And so actually we've studied, in the case of A-beta fibrils, the 40 residue A-beta peptide. So we've developed detailed structural models for 2 distinct fibril structures that are formed in vitro, and interestingly they have different symmetry. So one of them has a 2-fold symmetry axis about the long axis or the fibril growth axis. The other one has a 3-fold symmetry axis, and they're really quite distinct in terms of the data. They look different in electron microscope images. They have different NMR spectra when you isotopically label them and record NMR chemical shifts. They're different. If their mass density, mass per length is different in electron microscope measurements of mass density. So they really are physically distinct, and they propagate independently. If you prepare a pure sample of one, you can use that sample as a seed for growing subsequent generations of fibrils. And when fibrils grow from an existing seed, an existing fibril fragment, as the fibrils elongate, the structure persists along the length of the fibril. So the structure is self-propagating. So this is an interesting observation. It was surprising, but this is what the experiments led us to conclude. And so it was interesting to measure what is the difference in the growth kinetics and the differences in thermodynamic stability of the 2 fibrils. And so we did that in a paper that came out about a year ago in the Journal of the American Chemical Society. And it turns out the thermodynamic differences in stability are very small. Detectable but small, meaning on the order of 1 times kT or less. Small difference, and the stabilities are both very high. So if you take 2 different fibril structures and you separately put them in solution and let them sort of come to equilibrium with monomeric or soluble material, the solubilities that you measure are very low. They're sub-micromolar solubilities. So this means something. This tells you kind of why the polymorphism persists once it develops. So it persists because once you form a particular fibril structure, it essentially uses up all the monomeric peptide that's left behind. There's very little left, less than a micromolar under typical experimental conditions, and that's not enough. It's not a high enough concentration for new structures to spontaneously nucleate. So, once you have a given structure that's stable, it will just persist and will not convert to anything else on any reasonable timescale. On the other hand, you can get interconversion between 2 different fibril structures if both of them are present. So if you mix 2 different fibril structures, then you can see gradually the slightly more stable structures will increase in total mass, and the less stable structures will decrease. And this occurs not through an internal conversion, an internal structural conversion. It occurs as the less stable fibrils very slowly dissolve and transfer monomers to the more stable fibril structures. So that's the only way. Once you have a given fibril structure that's very stable, the only way it'll convert to a more stable structure is if that more stable structure is already present. And then there will be this very gradual, on the time scale of months or years or decades. It's very slow conversion. You can accelerate that conversion by breaking all the fibrils into short fragments by sonication or by some other procedure, because the overall time scale is limited by the number of ends, the density or concentration of ends of the fibrils, because that's where all the action happens. That's where the monomeric peptides or soluble peptides come on and off. So the more ends there are, the faster the whole thing will happen. So for us to actually observe it experimentally, which we did, on the time scale of a month, you could see that there was a partial transfer from one structure to another. So the more stable structures were increasing. But in order to see it, we had to break the fibrils into small fragments, and that you don't normally do that in experiments unless you're deliberately trying to do this. So consequently, the polymorphism, eventually, presumably there is a most stable structure for given conditions. It may be dependent on solvent conditions. But that most stable structure is not very much more stable than other structures. And the conversion to the most stable structure is very slow, because it depends on peptide monomers dissolving off of the ends of fibrils, which they only do very slowly. So I think that's sort of, I think by now we have sort of a reasonable understanding of the sort of biophysical or physical-chemical basis for the polymorphism, and it doesn't violate any fundamental principles. So it's basically a kinetically controlled system rather than a thermodynamically controlled system. Now the other interesting question is, why do the different polymorphs arise in the first place? And that's, so there we don't, we have only a qualitative understanding. I imagine what happens is that, for fibrils to start forming, well, you start with peptides that are initially, you can arrange, so initially the peptides are in a monomeric state, but then they will tend to aggregate quickly. They'll tend to aggregate. They form a variety of aggregated structures, some of which may be biologically important, the oligomers that are widely discussed. Other kinds of intermediates in the fibril formation process form under various conditions on various timescales, depending on exactly how you do the experiments. But then at some point, the oligomers of the peptide probably are somewhat dynamic within the oligomers, and then just by randomly they adopt a structure that is sufficiently stable that it will persist and start to grow. It's probably a structure that resembles the structure within the fibrils. We don't really know exactly what this critical nucleus structure looks like, but it would make sense if it kind of resembles the structure within the mature fibrils that grow from that nucleus. And apparently there's more than one kind of nucleation event. There's more than one kind of critical nucleus structure that is sufficiently stable that it will grow into a mature fibril. So there's just alternative nucleation events that lead to different fibril structures. So that's my understanding of it. But really, we don't know a lot about the actual structural properties of those nuclei. There are many paths. There's not just, there's two structures that formed in vitro that we've studied in detail, but we know from just looking at lots of samples that we prepared over the past decade, looking at NMR spectra and looking at electron microscope images of them, we know that there are many more than two, and other laboratories have identified other structures, and there's at least 5 to 10 of them, I would guess, each of which has its own signature in electron microscopy and solid-state NMR spectra. So there's multiple kind of nucleation events, apparently. It is surprising that there's such a diversity. It's an amazing thing. So that's seems to be true, but that was not predicted, I don't think. That was an unexpected result, that there would be so many of them. Well, so experimentally we found that fibrils that we derived from brain tissue of Alzheimer's patients those fibrils can have different structures than the structures that we've observed in vitro. They're qualitatively similar, but the details, the structural details are different. The precise conformations, some of the precise interactions, intermolecular interactions or interresidue interactions within a peptide molecule, those can be different. And why do we see different structures from brain tissue? Why do we see different structures from one patient to another? Again, it could just get back to this nucleation, that there's just a random nucleation event that determines what the structures are. In the brain tissue, it's also possible that, although in our experiments we extract amyloid from brain tissue of Alzheimer's patients obtained from autopsies, and then we use the amyloid fibrils from that tissue as the seed to grow fibrils that we can then do our NMR measurements on and electron microscopy measurements on. But it's possible that in the case of Alzheimer's disease is generally a slowly progressing disease, so there could be multiple structures that exist at very early stages of the disease, but by the time we actually get the tissue, after 10 years, or that maybe one structure has been selected out because it's particularly resistant to degradation or clearance, or maybe through this slow interconversion to reach the most thermodynamically stable structure, that by the time we get the tissue, it's actually come to equilibrium. I'm not sure I believe that, but it's possible. So we don't really know. But we have observed that the fibrils from brain tissue are typically structurally homogeneous within a given patient. We have seen exceptions to that, and so far the exceptions we've seen actually are from brain tissue from rapidly progressing cases. So in rapidly progressing cases, presumably the fibrils have not been present in the brain tissue for as long. So maybe this idea that there's a selection, and by the time you reach the end of your life, you've selected out a particular structure, that's possible. Alternatively, within that more heterogeneous mixture of structures that we've seen in a couple of cases, in rapidly progressing Alzheimer's cases, some component of that heterogeneous mixture may be particularly neurotoxic in a way that leads to the rapid progression. So we just don't know yet exactly what's accounting for our observations for fibrils derived from brain tissue. But this is ongoing work, so maybe we'll sort it out in the next couple of years. Yeah, I think that's the picture that I have in my head. That internally, internally the fibril core structure is very rigid. There's no opportunity really to actually rearrange. Internally involves breaking hydrogen bonds and rearranging the packing and the side chain interactions within. That's not going to happen internally on any appreciable time scale. So right, and unlike in the case of globular proteins, I imagine there's just the barrier to rearrangements, structural rearrangements, are not so high. Globular proteins, there's in many cases there's evidence that the proteins are continually undergoing structural fluctuations, transient unfolding events and refolding, and which allow them to kind of search the possible low energy structures and find actually the minimum, and apparently at least for proteins that have evolved functions, there's essentially one structure in many cases, in most cases, that is significantly more stable than others. Even within that most stable structure, there's always some intrinsic disorder, so it's never perfectly structured. There's always small variations, which maybe are underappreciated among protein biophysicists, but but they all look, you know, the structural differences are maybe not as dramatic as they are for amyloid fibrils. And so I guess in protein folding simulations that I've seen done by other groups, as a protein folds, it does get temporarily trapped in configurations that are not the native structure. You know, it can get trapped in configurations where it has a lot of the native secondary structure but doesn't have the correct tertiary contacts, and those will persist, but they'll persist maybe for microseconds on that kind of time scale, and then there's just enough thermal energy to rearrange things eventually. So in the case of the amyloid structures, they get trapped forever because the kinetic barriers are too high. Just this very extended supramolecular structure that it has, so you can't, yeah, without disrupting the whole thing, you can't just rearrange one little part of it. Yeah, so the question is, can we use our structural models as the basis for docking calculations or for the development of inhibitors? Or I also think an important target is imaging agents, so developing compounds that bind selectively to perhaps the particular structures that are most significant in the disease. That could turn out to be important. But do our structures have sufficient precision for that to be done? I'm not sure. When I describe our structures, I refer to them as detailed molecular structural models because within the field of structure determination, if you say you have a structure, people then they interpret that in a particular way, and I want to avoid getting into arguments with people about, do we really have a structure, or do we have a model? I mean, I think all structures are models at some level, and it's just a matter of degree, and it's a continuum and all that. I don't think there's a real threshold that separates detailed structural models from structures, but I don't want to get into that argument because I think it's pointless. But from a practical standpoint, do we have enough precision in our structural models to use them to actually differentiate or to calculate binding constants of different small molecules to the fibril structures? I would say unlikely, because there are actually certain aspects of the structure that are essentially impossible for us to determine experimentally. So like in any structure, well, I won't say any, but in many structure determinations by various experimental methods, I mean, in reality, there are certain aspects of the structure that are known very definitely with high precision, and there are always other aspects that are less certain. And that's definitely true of our work. One of the particular aspects that's very difficult for us to sort out has to do with something that we've called staggering in our papers, which is when you have an amyloid fibril structure. These are primarily beta sheet structures with a cross beta orientation, so ribbon-like beta sheets running the length of the fibrils. But when you have a situation where there are multiple layers, and in particular when each molecule contributes to more than one layer of beta sheets that are stacked on one another in some way, then there can be a shift of the top layer versus the bottom layer in one direction or the other, so that they're not specifically the beta strand segments that arise from a given molecule don't necessarily sit on top of one another. They can be displaced in one direction or another. And when we've looked for that, which we can do by isotopic dilution experiments, when we look for that, we inevitably have found that, in fact, the contacts between beta sheet layers indicate that they are shifted or staggered in one direction or another, and we don't know which direction. We have no way of telling whether it's in the two ends of the fibrils are actually structurally inequivalent, so they're shifting in one direction is different from shifting in the other direction. And we can't determine that from our data. So that's one very specific aspect of the structure that's really indeterminate. And so then you'd have to, of course, then, if you wanted to try to use the structural models to design compounds to inhibit them or to dock to them, or then you'd at least have to consider those two possibilities, or maybe it's more than two. And then there are other aspects of the structures where we don't really have sufficient information about the precise aspects of side chain conformations and things like that. So they're not at the level of a 1.0 angstrom X-ray crystal structure by any means, and I would not make that claim. But I think our structures are what you see. Sometimes when I give talks, what I tell people is, like, if I show the structural model on the screen, I say, well, what you can see from the back of the room is certainly correct. If you're sitting in the very front row, some of the details, may not be correct. And also when we've deposited the coordinates for the structure in the protein data bank, I've tried to do this in a way that's as accurately as possible reflects the uncertainty in the structure. So there's bundles of structures that have disorder, structural variations, and I've tried to, rather than minimize that, I've tried to maximize that. So I've tried to include as large a range of structures that actually fit our data equally well or sufficiently well in those structural bundles. And if you look at those, you'll see that if you actually look at what's been deposited in the protein data bank, you'll see that there are significant structural variations within the bundle of structures, and that gives you a reasonable picture of what we actually know and what we don't know. Now, another perhaps more fundamental question is, if you want to inhibit the formation of a fibril, what do you need to do? So the fibrils have a given structure along their length until you get to the ends. And then the ends of the fibrils, presumably there's some distortion of the structure. And if you want to cap a structure or prevent its propagation, it seems to me you probably need to know something about what's happening at the very ends of the fibrils. And that, we can't do that. We don't, I can imagine futuristic ways of doing that, and maybe that'll come up later in the discussion, but at this point we don't really know how much of a distortion of the structure exists at the ends. But there has to be something, because obviously there's kind of a dangling hydrogen bonds or dangling interactions at the ends, which have to lead to some kind of a distortion. More in the 2 to 2.5 range probably, or something like that. So, we can calculate, though, these RMSD values from our structural bundles, and you get numbers in the 2 to 2.5 range, I think, for the I think of 4 angstrom structures is. But again, there's certain aspects of it, there's certain parts of the structure that are 4 angstroms or something, but then there are other aspects that are much better. So it's hard to really, a single number really can't capture the whole thing. So there's a question of how much intrinsic disorder is there also. So and that may vary from one fibril to another. There are certainly the quality of the solid state NMR spectra, the sharpness of the NMR lines, which is some measure of the level of disorder, although it may be difficult to quantify. But certainly that varies from one, even for the beta amyloid fibrils, it varies from one fibril type to another, and certainly from one fibril forming protein to another. There are some that give spectacularly sharp solid state NMR lines, which must mean two things: that they basically have a very well-defined structure, and then they also have internal dynamics that average out the inevitable rapid internal, so all the motions that are happening on a short time scale, very, very rapid. So those two things have to be true, I think, to get very sharp lines. In other cases, we just never see lines that are quite that sharp, and there's some evidence that there actually are, that there may be intrinsic disorder along the length, even within a single fibril along its length, there can be some variations. which is going to limit. But it's these are hard to quantify, these things. So I should a point that I like to emphasize is, of course, when we started on this work, which was now about 15 years ago, very, very little was known at all about the structures, and so one should not set an artificially high bar for the work. I mean, starting from really zero or very little, I think we've come very far. Not just us, but the field in general has come very far, and these are difficult problems, and we've had to figure out a lot of things along the way, like which measurements work, which measurements don't work, what's the right combination of measurements that allow you to get to a structural model, a full structural model, and how do you actually make the samples? This issue of polymorphism, we weren't expecting that to be a problem when we started. We didn't know. So it's how far will there certainly are certain cases where the solid-state NMR spectra are extremely clean, and you can actually, and the monomer is not too large, and you can actually isotopically label it the way you need to do for the measurements. You can actually make the samples, and then you can get very far, and you can get quite precise structures. And there's other cases where that's just hard to do. It's just very hard to do, but I think overall, our understanding has improved quite a bit from where it once was. Can you crystallize fibrils? Obviously, other groups, principally David Eisenberg's group from UCLA, has had spectacular success in determining very high resolution crystal structures for amyloid forming peptides, and in those crystal structures, the structure is a cross beta structure that is, if not the same, is very closely related to the structures that one observes in amyloid fibrils. And there's the same issues of polymorphism that they observe and various structural variations. An amazing thing about those crystal structures is that the beta sheets are absolutely flat. So to actually crystallize, you have intermolecular beta sheets that are untwisted. Otherwise, you wouldn't have the periodicity that you need to have, the translational symmetry that you need to have in a crystal structure. Fibrils do tend to twist. Beta sheets in proteins tend to twist. So far, that seems to probably be what's limiting the ability to actually crystallize full-length amyloid-forming peptides or proteins into their amyloid-like state. So far, if you ask what's the real barrier to crystallizing them, determining crystal structures, I think the twist, the inherent tendency for beta sheets to twist, makes it difficult to crystallize. Of course, with crystallography, you should never say it's impossible, because eventually they succeed. As numerous cases, numerous examples have shown, you should never count the crystallographers out, because they always figure out a way to do it. And then it's spectacular, and they can actually get all the details. It's a great technique. So far, it hasn't worked for large peptides or proteins, but maybe it will. Maybe it already has, and we just haven't heard about it. I don't know. But then, even then, there's the issue of the polymorphism. So then, which of the structures, there are structural variations, and which of those will turn out to be biologically relevant still remains an open question. So then, so our experiments where we're trying to actually characterize the structures that actually develop in tissue, that's something that may be hard to actually apply crystallography to that. And other groups are doing very similar things now for other amyloid-forming proteins, alpha-synuclein, for yeast prion proteins, maybe eventually the mammalian prion protein, using extracts from cells or from tissue, and trying to, sort of then focus in on the biologically relevant polymorphs. That's definitely the trend now in amyloid work. Yeah, okay, so it's conceivable that you could, in the twist, because that's kind of a soft degree of freedom, in electron microscope images, some fibrils obviously have a twist, but it's somewhat regular in some cases, but it's not perfectly regular. And this is actually, I think, made it also difficult for electron microscopy to get real high resolution results from electron microscopy. That plus the polymorphism again. But as I said, yeah, you can't rule it out. You can't rule it out. Somehow, eventually, someone will figure out a way of making it crystallize. Well, it's certainly amyloid formation is a very prevalent phenomenon, and it sort of seems that something that proteins or polypeptides like to do if they don't have something else to do. If there's not a more stable, monomeric or oligomeric state, then this is something they tend to revert to, because and when you look at the structural models, you can see why they are kind of good, happy structures for polypeptides. There's all the backbone hydrogen bonds are formed. The residues are in the beta sheet, beta strand kind of region of the mitochondrion, plain mitochondrion space, which is accessible to all amino acids, and it's a place they like to be. Avoids steric clashes among the side chains, so you can sort of understand why many different sequences would sort of fit into an amyloid structure. The most common structure that seems to occur is the parallel beta sheet, in-register parallel beta sheet structure, which was first seen by David Lin and Steve Meredith and Bob Botto in their experiments on the residues 10 to 35 of the A-beta peptide. They found the first evidence for in-register parallel beta sheets in amyloid fibrils, and that was very controversial when they reported that in 1998, but that turned out actually to be correct and actually the most common amyloid structure. And that's a structure that automatically brings within one beta sheet. It automatically lines up the hydrophobic residues with one another. It automatically lines up polar residues, glutamines and asparagines that can interact through their side chains in a favorable manner. It also brings charges. It creates rows of charges, which is bad, because if you have, in a parallel structure, if you have many charged side chains all lined up about 5 angstroms apart, if they're in the interior of the structure, and if it's a fibril that particularly has a hydrophobic kind of interior, so it's a low dielectric environment, then the electrostatic repulsions there would easily disrupt the structure. So that doesn't happen. So for a polypeptide to form an amyloid structure, it has to avoid creating these rows of like charges in a hydrophobic interior. And so you can prevent it if you have a sufficiently highly charged sequence. It's also okay if the charges can pair up, but if there's a net charge, if it's a sequence with a large net charge, that's one way to prevent amyloid from forming. If there's lots of prolines, that will prevent amyloid from forming. But otherwise, most sequences, it's something they seem to be able to do. And so that's why it's such a common occurrence. Yeah, there are some amyloid-like structures, cross-beta type structures in real proteins, and then there are various functional amyloids that have been either proven or at least postulated. Well, I can't say very much about functional amyloids. There are several cases where it's documented that the amyloid state has a biological function. Some of these, and I'm not really the expert on this, but some of these are have sort of an adhesive function. So the curly proteins on the surfaces of bacteria act to kind of at least allow the bacteria to adhere to surfaces or to one another. There's, in the case of the het-S fungal prion, which is a very remarkable protein that's not polymorphic, and for which the solid-state NMR spectra originally obtained in Bayat-Meyer's group at the ETH in Zurich. I mean, they're just spectacular, fabulous spectra, and as a result of their hard work and also the favorable properties of the protein, they've developed a very, very detailed structural model for those filaments. So those are believed to have a biological function in what's called heterokaryon incompatibility between two different fungi. Yeah, so the question is about the future of solid-state NMR. So of course, solid-state NMR is the main experimental technique or set of techniques that we've used to characterize structures of amyloid fibrils. And this is overall applications of solid-state NMR to biological systems is an area that's grown quite rapidly over the past, let's say, 15 years, something like that. And there's been a lot of progress, and I think so. A lot of the progress has just been because there are now a large enough number of groups who are just trying things. So I think what's happened, one of the things that's happened is in solid-state NMR is that people have been able to find classes of systems where the solid-state NMR techniques really work, and you can get high-quality data. You can get sufficiently resolved spectra, and you can actually make the samples and get sufficient sensitivity. And the techniques have also come along at the same time, but I think just exploring different applications has been an important part of it, and will remain an important part of it. I think it's still that remains to be done. I think and I don't know what those in the future, in the coming 5 years. I'm sure people will find more and more classes of systems where the solid-state NMR methods really work, and where you can learn things that you can't learn from other techniques. And in principle, solid-state NMR is great because you can use it to get atomic level structural constraints, structural information about systems that you really can't study in detail with other techniques because they're not soluble, or you can't crystallize them, or they're too large for your system, for your technique. So in general, in very general terms, it's a technique that should have very wide applicability. But then you also have to find specific systems where where you can actually make the samples, and where the solid-state NMR spectra are sufficiently simple that you can actually understand them and get adequate sensitivity. And that's kind of, we have to identify more of those. And so some of those, so the amyloid work that I've been involved in is one good example of that, but there are other examples that have come out and will continue to come out, I think, from a variety of groups who are working on this now around the world. And it is still a growing number of groups, and so that's a very healthy thing for the field. I think when I think about what's the big limitation, a big limitation is really on the biological side. So we're limited in terms of this complexity and the size of the systems we want to study. We have to isotopically label the protein. So far it's mostly protein work. You'd like to be able to look at proteins that have molecular weights of 100 kilodaltons or more, and in principle you can do that, but the spectra end up being intractable because there's just too many signals if you isotopically label the entire sequence. And so really segmental labeling techniques, so molecular biology techniques, protein expression and labeling techniques, and ligation techniques that allow you to really prepare, really on the scale, the quantities that you actually need for the measurements, which is at least typically milligrams, to actually make proteins where you've just labeled a subset of the amino acids in a particular segment of the sequence. My group has worked on that continues to work on it at some level. Many other groups are working on it because it's obviously what needs to be done. And hopefully that will be worked out. And once that, if that can be developed, just the sample preparation side, the labeling side, if that can be made into a general kind of technique that allows you to get kind of milligram quantities of proteins that are segmentally labeled in a general way, then that would open up all kinds of new applications for the technique. So that's really probably. And then there's always ongoing just NMR technique developments that are happening all the time. Pulse sequences are getting better. Sensitivity improves. Proton detected carbon-13 and nitrogen-15 NMR in solids is something that we worked on a while ago, and other groups are doing it a lot now. And sensitivity is getting better as a result. Magnetic fields are getting higher, etc. So the sensitivity, I think, is there, and it's just the complexity of, and the sensitivity is there, and the methods for actually getting the structural constraints that you need. Those continue to get better, but they exist already, and really making the samples, making large proteins that are segmentally labeled would actually very quickly allow you to do all kinds of things that you can't do now. So that's the breakthrough that's needed, I think, more than anything. On the sensitivity end, there's this dynamic nuclear polarization technique, which has become very popular, and my lab is heavily involved in that also. For solid-state NMR currently, the dynamic nuclear polarization involves introducing paramagnetic dopants into your sample, carrying electron spins, which are intrinsically more highly polarized, have bigger differences between population differences between spins up and spins down in the magnetic field. So they're more highly polarized than our nuclear spins by a factor of 660 for protons. And then transferring polarization from the electron spins to the nuclei, thereby enhancing the polarization of the nuclei and enhancing the NMR signals proportionally. So those techniques are now becoming more and more widely used. Currently in solids, those are experiments that are done at low temperatures, because the mechanisms that work best for dynamic nuclear polarization in solids are mechanisms that are effective at low temperatures. So going to low temperatures is a good thing in terms of sensitivity, but it generally costs you a lot in terms of resolution. So the NMR lines get broader at low temperatures because you just get all the motions that tend to average out structural inhomogeneities get frozen out, and then you're stuck with sort of static structural inhomogeneities that lead to broader NMR lines. So you sacrifice resolution, and the amount that you sacrifice seems to vary a lot from sample to sample, but it's always there. It's always significant, and so that depends on exactly what temperature you're working on. So some groups are trying to do the DNP at higher temperatures where there's less of a line broadening effect. So that's that's something that can allow you to look at certain problems that are otherwise difficult to look at. So that kind of breakthrough in sensitivity is important, and I think where it's obviously important is if you're looking at molecules that do not have a large number of isotopically labeled sites. So the spectra are inherently relatively simple, and then you can sacrifice resolution and still get meaningful results. So if you can, again, selectively label or segmentally label in relatively short segments a large protein, or in our case, amyloid-forming peptides that we can synthesize, directly synthesize them chemically, and then we can isotopically label specific residues or specific segments. So then the DNP experiments are, can be very useful for those kinds of systems. Ligands, so hormones that are relatively short peptides or other small molecules bound to receptor proteins, and then you're interested in what the ligand structure is in the bound state. Those kinds of problems, I think, are perfect targets for dynamic nuclear polarization experiments. And then that overcomes the problem of, in the case of hormone receptor complexes, getting the receptor protein is usually the limitation. You need, the receptor proteins are typically integral membrane proteins that you just can't prepare in most cases in large quantities, so then you have a sensitivity issue there. And then with DNP you can overcome that, and then you can solve problems that you can't otherwise solve. So that's another thing that's coming along now, and probably in the next 5 years we'll see a lot more of that. So those are at least several of the areas. Question about synergies between different experimental methods, and I think it's very important. And in my own lab and our own work on amyloid and other systems, we're working on viral capsids, proteins, and the assemblies that they form. And electron microscopy is just invaluable. You can't live without it, really. It does, before you do the NMR measurements, you have to be able to see the structures, see the assemblies, and make sure it's what you think it is. Otherwise, you can very easily go astray. And this polymorphism that we see in fibrils, you have to. The electron microscopy gives you, pretty strong evidence that you have a sample that's morphologically homogeneous, and so that's very important. We use atomic force microscopy to some extent for similar purposes. I think so. Other techniques that might, well, certainly calculations. So molecular modeling, molecular dynamics simulations. That's so computational studies are certainly important, are an important component of any of these studies. EPR, electron paramagnetic resonance measurements, can provide information, structural information on a different length scale, on a longer length scale than what you can readily achieve from solid-state NMR. So that's an obvious combination that NMR people in general are taking advantage of now in their studies, and you could then use also optical techniques, FRET techniques. As another way of getting long-range distance information, and probably others. So obviously, we rely heavily on mass spectrometry. For some of our problems now, we're trying to combine solid-state NMR results with hydrogen exchange, hydrogen to deuterium exchange measurements that are detected through mass spectrometry. So that can be can give you information that is very helpful in identifying in a fibril forming system the fibril core from the material that's outside the core. That's what we're trying to use it for. So that's just a few examples. There are probably others that I'm not thinking of, but I think at one time I know my own training in NMR. When I was a student, the idea was that you were an NMR expert, and so you should know NMR, and but you don't have to know anything else. And in fact, the tendency was to avoid learning other techniques, and part of that was that I think a psychological effect that when you become an NMR expert, you really understand NMR in really great detail, and there's a lot of the mathematics behind it. There's that's kind of what attracts us to the field in the first place. In some cases, has a very interesting mathematics behind it, and there's just a lot of details to do the NMR experiments themselves. You have to know a lot of details. It's a field of details, and then you go to some other kind of technique where you're really a novice. You don't know very much about it, and you feel very uncomfortable because you're used to working. To doing measurements where you really understand everything about it. You understand exactly how the equipment works. You could build it yourself if you had to, and you understand really all the physics behind it, all the mathematics behind it. You understand it in great detail, and then you go to do electron microscopy, for example, and well, you're not really so sure how the machine works, and so you feel like you're cheating kind of by doing that, or But that's something you need to get over, I think. But that's kind of been a barrier, I think, for NMR people, is that they kind of, this familiarity that you have to kind of overcome. If you do another measurement, and you have to live with the fact that maybe you're not going to be such an expert on it, but still you should do it, and it's still very valuable. And if you don't do it, then your NMR measurements are not going to be as meaningful.